Hi, my name is Lei, and I will give a brief introduction on our latest work, which is a comment on the misuse of reporter score in microbial enrichment analysis. So the enrichment analysis has become a common technique in bioinformatics studies, and many methods have been proposed to facilitate the biological interpretation of expression data. Recently, a novel method called reporter score has been introduced into microbial research for pathway enrichment analysis, and we noticed that in some of these studies, reporter score shows the capacity of not only calculating the enriched pathway, but also using it for determining the regulation direction. However, after our careful review, we found that these studies used reporter score for the wrong purpose and misinterpreted the results. The reporter score algorithm was first developed for identification of reporter metabolites in metabolic regulation. It converts p-values of enzymes to z-score and aggregated them by metabolite and enzyme interactions. In recent years, researchers have migrated the reporter score algorithm to microbial enrichment analysis. However, as we said, we found that some studies misuse the reporter score. From Formula 1 and normal distribution, we can see that z-scores monotonously decreases with p-value increases, and the negative z-score corresponds to non-significant differential genes with p-value greater than 0.5, and thus it means the larger the reporter score, the more significantly enriched the pathway, while a negative reporter score indicates that the gene set is not enriched in this pathway rather than the pathway is downregulated, and therefore it is obviously wrong to regard the plus or minus sign of the reporter score as regulation direction. To illustrate the idea, we created a set of 200 KOs and 10 pathways as simulated data. Among them, 50 KOs were significantly differentially expressed, and to better explain that the reporter score is non-directional, we artificially set that KOs in the same pathway have consistent regulation direction. According to the results, the pathway 4 with the lowest reporter score was predominantly made up of the least significant KOs, which notably is independent of the expression directionality. However, if we use a cutoff of absolute 1.64 that is commonly used in surveyed articles, pathway 4 will be misinterpreted as a significantly downregulated pathway. Needless to say, misinterpretation of the reporter score will cause misleading results in pathway enrichment analysis. And compared with hypergeometric tests, the results of reporter score showed similar trends, and the pathway with higher reporter score were also marked as enriched by hypergeometric tests. This reporter score was first misused for microbial enrichment analysis in 2015. Similar misuse has been found in at least 45 papers, and the influence of these papers showed an exponential growth trend. Most striking, as far back as 2008, the two authors of reporter score proposed a workflow called reporter feature analysis, and by using it, the regulation directionality and the statistical significance can be determined simultaneously. This workflow has been applied over a broad range of scientific topics, including microbial research. In our demo dataset, reporter feature achieved the results of up and down regulated enrichment, and it is an important complement to reporter score. The reporter score does not take direction into account. For example, the two pathways 2030 and 2040. They ranked high in reporter score but were significantly downregulated, and the pathway with reporter score greater than 1.64 can either be upregulated or downregulated. So in this command, we want to raise awareness that in scientific research, special attention should be paid to the misinterpretation or misuse of statistical techniques. And we found that interpreting reporter score under habitual thinking and adopting a published method on face are the two underlying causes why such a naive error kept being made. And in the paper, we also give some suggestions to promote rigorous research and to guarantee the significant and stable development of bioinformatics. So this is a brief introduction of our paper. If you have any questions, please feel free to email us. Thank you very much.